Hello guys, welcome to IB 360. So today's lesson we will talk about nucleic acids. So nucleic acids are long chain macromolecules which are formed by end-to-end polymerization of large number of repeated units called nucleotides. So in this statement there are many terms which you might not be aware of. For example, what are nucleotides or what is polymerization? What is a macromolecule? So a macromolecule is basically a very large molecule, like protein, which we had talked about earlier, was a macromolecule. So nucleic acids are also macromolecules. They are long chains, and that means they are hugely sized molecules found inside our body cells, and each and every living organism has a genetic material inside them. And for human beings, the genetic material is DNA, which is deoxyribonucleic acids. We also have another type of nucleic acid known as RNA or ribonucleic acid. Both have similar structure, but there are certain subtle differences which creates huge impact in their functionality. We will study about them today in this video, but first of all, let us talk about the general structure of the nucleic acids. So as you can see that it is a helical structure. Nucleic acids are helixes. So there are two strands which are running against each other, which are, we can say, anti-parallel to each other. These two, this is one strand, another would be this. So these would be two strands which are running opposite to each other. So why opposite? Because they have opposite polarity. If I call this this strand, this A, and this strand as B, if A runs from 3 prime to 5 prime, B will run from 5 prime to 3 prime. That is why anti-parallel. But where do these numbers come from? 3 prime or 5 prime? That you will understand when you understand the structure of the nucleotides of these nucleic acids. Anyway, so nucleic acids, they show a wide variety of secondary structures. Alpha helix of protein was a secondary structure, if you remember. So by that you can easily memorize that it's a secondary structure. A wide variety of secondary structure. Okay. So as far as DNA is concerned, DNA is our genetic material. That means offsprings, they carry their parents' uh, traits via the DNA. The DNA is the one coded information stored inside the nucleus of every cells, which pass on from generations to generation, thereby thereby conveying our traits, our genetic traits, and establishing the entire concept of heredity or inheritance. Okay, so it carries all the hereditary information coded in the arrangement of its nitrogen bases. Okay, now the thing is that DNA also has another property of replication, which is an autocatalytic function, and that is the primary function essential for passing the genetic information from one cell to its daughter cell or from one generation to the next. Okay, so this is a very important trait of DNA, and that enables it to be a very good genetic material, because it can convey itself to other cells. And remember one thing which I said, that the coded informations are in the form of in the arrangement of the nitrogenous bases. So without mincing word, let me give you a clear concept of its nucleotide. So see, these four boxes which I have made, these represent nitrogen bases. These represent nitrogen bases, and there are 4 types of nitrogen bases in DNA. See, adenine, guanine, cytosine, and thymine. So these all are cyclic compounds. What are nitrogen bases? They are cyclic compounds. Okay, and they have nitrogen in them, so they are known as nitrogenous bases. And see, these are their structures. This is cytosine, this is guanine, this is adenine, and this is thymine. One thing which you must have noticed is that both guanine and adenine, these two nitrogenous bases, they have 2 rings. So they are called purines. While cytosine and uracil in case of RNA or thymine in case of DNA, both, they would be called pyrimidines because they have single ring. So one is the purine, one is the pyrimidine. New terms which you are knowing now. These two very important because you see single ring 1 and here also single ring, but here 1, 2, here also 1, 2. So these are supposedly a schematic structure of the nitrogenous bases. So let me say this is adenine, and let me say this is adenine. Okay, and this is okay, let this be cytosine. So this will be thymine and this will be guanine. So see, adenine always establishes bonds with thymine and guanine always with cytosine. So it will always be ATGC and never AG or CT. This will never happen. So adenine with thymine, it will establish the bond via 2 very good looking hydrogen bonds, and cytosine with guanine will establish 3 hydrogen bonds, or vice versa. And these are the nitrogenous bases. These are the hydrogen bonds, these lines, and they are then attached to their pentose sugar individually. These are the pentose sugars via glycosidic bond. I'll write the name of the bonds again, so no need to worry. And then they are linked via phosphate group. Okay, this is what is happening always. You see, these are the base pairs. These are the base pairs, and this is the strand which we are talking about is basically this part. This part. This part is a strand. And one phosphate, again this is another with another phosphate, this will be linked. And this will go on. Okay. So one nucleotide is basically one nitrogen base plus one pentose sugar plus one phosphate. This is one nucleotide. Okay. This is one nucleotide. One nitrogen base, which is this one. One pentose sugar, which is this one. And one phosphate group, which is this one. So this trinity is this trinity is our nucleotide. This trinity. Okay. So as per the definition of a nucleic acid is concerned, it is formed by end-to-end polymerization of large number of nucleotides. These nucleotides, they are repetitive, and polymerization is established by this phosphate bond. It is a diester bond because you see there are 2 bonds. It forms one with the lower pentose sugar and one with the upper pentose sugar. That is why it is known as a diester bond. And this results by elimination of water molecule. Here also water molecule is released, and this is N-glycosidic bond. It is a NC bond, okay, formed between nitrogen and carbon. Nitrogen of the nitrogenous base and one carbon of the pentose sugar. Likewise. And these are obviously hydrogen bonds. Fine. Another term which often comes as a question is, what is a nucleoside? This is a nucleotide. What is a nucleoside? So nucleoside is nothing but the nitrogen base, the pentose sugar, and that's it. No phosphate group. Only so only this will be a nucleoside. Only this, the nitrogen base and the pentose sugar, and not the phosphate. That will be the nucleoside. Okay. So let us get back to the diagrams. Here you see the anti-parallel strands. They're parallel but they're in opposite direction, so they would be called anti-parallel. And DNA is circular in case of prokaryotes. Okay, in case of prokaryotes, like in case of bacteria, DNA is not linear, it is circular and complete. And in case of bacteria, their one chromosome is formed by the entire DNA, obviously, so they don't have multiple chromosomes like in eukaryotes. In eukaryotes, we have, in case of humans, we have 23 pairs. In somatic cells, we have 23 pairs of chromosomes, which means total 46 chromosomes. So total 46 DNA macromolecules. Okay, total 46 DNA macromolecules which are coiled to form individual chromosomes, highly coiled. And for that coiling, many packing proteins are required. Histones are famously known as the packing proteins in case of DNA. Actually, in this DNA packing and all, you will again learn about these things in translation chapter. But these are very basic informations which you must know. One more thing is, it is also known as a duplex, DNA duplex. Okay. And another bit of information is that the distance between each base pair. The distance between each base pair, that means if this is a base pair, that means these are two nitrogenous bases which are bonded through hydrogen bonds. So this distance is 3.4 angstrom. Okay. This distance is 3.4 angstrom. Very, very important. And the anti-parallel thing which I was mentioning at the very beginning of today's video, the 3' and 5' thing, where does this come from? So see, in one of the chain is running in 5' to 3' direction, while the other one is running from 3' to 5' direction, right? And this is what is complementarity, which we say. This thing is complementarity. They complement each other. If we look back in the nucleotide structure, we see that the phosphate end, that means this end, this phosphate end, this has a free end at 5'. That means the pentose sugar, this carbon which is binding with the phosphate, this is the 5 carbon of the pentose sugar, 5 carbon. Okay. And the other end which remains not bonded, that is the 3 carbon of pentose sugar. So the ends which remain unbonded, they would be 5 carbon and they would be 3 carbon. So from there, see, if you have any problem, you see, let me. So see, this is the 5 carbon, and here the pentose sugar is again bonding with this end. So here whatever is, whichever carbon is left unbonded is the 3', and here it's the 5'. So thereby this 3' and 5' establishment comes into play. Okay, because the direction in which they're moving is absolutely opposite to them, and that establishes anti-parallelity, and that is what is the main reason for their complementarity, that they are complement to each other. The two DNA chains are not identical with each other, they're in opposite directions. And also, uh, if one is A, another must be T. If one is C, another must be G. If one is again G, another must be C. If another is T, one is T, another must be A, and accordingly they have to proceed. So this is the main reason behind the complementarity. There is also another thing which is known as sense and antisense strands. See, both the strands of DNA, they do not take part in controlling heredity and metabolism. Only one of them does so. Okay, the DNA strand which functions as the template for RNA synthesis is known as the template strand or the antisense strand. Okay, so one of them will be the uh, template strand, and this is the antisense strand, and its complementary strand is known as the non-template strand or the sense strand. Okay, so this is a very important concept. RNA is always transcribed on 3' to 5' direction of the template strand of DNA, or we can say in 5. From, this is the direction of transcription. This is the direction of transcription, and this is the strand, the polarity of the strand which gets transcribed, the the antisense strand or the template strand. Okay, so I hope you understood today's lessons. If you have any doubts, you can leave them in the comment box. I will try to address them. Till then, stay safe. Thank you guys.